you for joining us. For those of you who don't know me, I am Dane Kreiner, the Executive Director of the Center for Science, Technology, and Society here at Santa Clara, one of our 3 centers of distinction that embody the university's mission to create a more just, humane, and sustainable world. We are delighted to welcome Drew Endy, who is a leader in the field of synthetic biology, to Santa Clara today. Drew has degrees in civil, environmental, and biochemical engineering. Did postdocs at UT Austin, one of my alma mater, University of Wisconsin-Madison, and then joined the MIT faculty in 2004. In 2005, he formed the BioBricks Foundation, and by way of full disclosure, I am on the board of the BioBricks Foundation, and Holly Millian is the executive director of the BioBricks Foundation. So if you're interested in learning more about the great work, which is really focused on engineering of biology in a way that is open and ethical to benefit all people in the planet. Drew came out to California, to Stanford in 2008 in the bioengineering department, and we are especially happy to have him here today because he just came back from the Netherlands. The plan for today, he'll talk for about 45 minutes. We'll have an open Q&A for about 15 minutes, and then at least for my Technology for Social Justice class, Drew will stick around and we'll have an open discussion about many of the issues we talked about in class earlier today. So without further ado, please welcome Drew Endy.
Thank you so much. This is a new talk for me in that I'm really hoping we get to a conversation about ways in which biotechnology could be meaningfully distributed beyond current modes of organization, current modes of concentration. And to be frank, I don't know if that's possible, good, bad, dumb, dangerous, or some combination. So I'm hoping that I'll actually finish before 45 minutes, maybe 30 to 40 minutes, and we can talk about it. So I'm going to try and leave an impression, but you can teach me from what I say where we might want to go instead. I understand there's some folks here who are studying ethics and technology, and I want to acknowledge that I've yanked a lot of slides I usually put in my talk having to do with ethics and technology, which is fine because we probably have enough time, and if you want those, ask a question and I'll pull them up. All right? So the talk here is organized into 4 sections: dreams, realities, tools, and opportunities. And the bookend of dreams and opportunities is where I really want to focus your attention. So if you just pay attention to the first slide and the last slide and daydream in between and then come back to the conversation, that'd be fine. But I'm really trying to think about how we might partner globally to do more with biology. So here's some examples of dreams I naively wonder about. It seems to me that you can do a lot by using existing distributed modes of biomanufacturing, the making of bread or yogurt, and reprogram that locally to make things that are needed. I have this fantasy. I'm thinking I should refocus my lab to program an algae to grow into a biofilm that would take in photons and do reverse osmosis to provide distributed manufacturing of water via a reproducing machine. It's technically crazy because you don't have active transport of water molecules in ways that we know about. But this is the dream section, so I get to the dream. Agricultural biotechnologies that are responsibly sourced and perhaps community-owned. Imagine full-scale genetic engineering of crops for foods and fuels, hugely controversial, but controlled by the people who are doing the farming. Microbes and plants that detect and remediate environmental pollution. An improved global understanding of nature through distributed and equitable access to living technologies. And from that understanding, hopefully an improved partnership. So here's some dreams. I wanted to share some dreams that came from the plane over Greenland this morning. Realities. I don't do any of these things that I'm dreaming about right now. I work on fundamental tools which are typically invisible that, as they get better, change the process by which other people partner with biology to make stuff. So let me give you a sense of how lame and beautiful these tools are. So for example, we have a paper coming out next week, which is the first major paper since we moved the lab to California, And it's on this area of bio bits, not bricks, but biological bits, or genetically encoded data storage. If you think of a USB memory stick, that's nice, that's a gig of bytes. I want 8 bits, I want 1 byte. And Jerome Bonnet from France, together with Pakun Sukorn from Thailand, have been the two really making this work. So what they figured out how to do is to take a chromosome inside an organism and design a specific sequence of DNA that can be targeted in such a way that you could change the orientation of this element, which we call the Flippy. So the Flippy gets flipped. And you can flip it from one orientation to the other, and from that orientation back to the original. So it doesn't take too much to see that if you can do this, you could declare that if the Flippy is pointing left, that could be a 1, and if it's returning to its original state, that could be a 0. Now we have a binary digit data register, or a bit, encoded in the chromosome. In order to make this an engineered system through which we can store extrinsic information, like the amount of pollutant in the environment or something like that, we have to be able to do this flipping. And to do that, we use enzymes. The set enzyme is called an integrase, redeployed from a virus to do something a little bit different than what it does normally. And this is what it looks like. We can take cells in a 0 state that are green. We give them a little bit of pulse of the integrase. They turn red. And when we remove that input pulse, they stay red. So they've kept a memory of the input pulse. We can make this input pulse anything we want. So it's a generic data register. You could store information about anything that could turn on gene expression. To make this useful, in a scalable sense, we have to flip it back. To do this, we take the same protein, we express a second protein, that's it, and that should change the direction and flip it back. Get us back to 0. So we have cells that are red, we give them both, they go green, and they mostly stay green in this experiment. You can see one cell lost its memory, right? It got corrupted. That'd be called a spontaneous switching event or something. So we can mostly erase. All right. That was all working 3 years ago. So then took a whole bunch of work to figure out how to get the reset and the set working together. What does this have to do with making the planet better? Right? I'm just trying to give you a sense of who I am, and we'll get back to that. For example, if you run this reset operation and that excisionase accidentally goes away first, your reset becomes a set, and that's a disaster. Right? You fix that by making a lot of this protein and stabilizing it so it doesn't degrade. But when you do that and you put it in a cell, that protein that modifies the activity of the integrase is always around spontaneously, just to the noisy, spontaneous differences in behavior. So that corrupts your set operation, turning that into a reset. So that's broken. How do we fix this? We degrade the protein faster. We underexpress it, all the things that were leading to the original problems. So we really got into a lot of trouble. And when we debugged our trouble, we came to the conclusion that the genetic elements or tools that we were working with weren't well engineered in the first place. That we were trying to build something very precise, but we were using found objects from nature that weren't themselves designed to be easy to put together and get precision behavior. Thus, 3 years go by, and Jerome and Pakun spend 750 design-build test cycles in the laboratory, designing different DNA sequences, constructing the DNA, putting it into cells, seeing what happens under the microscope, and they get it working. So we now have what we call a recombinase addressable data module, or RAD data module. I grew up skateboarding, so it's really exciting when you have a paper coming out that says RAD. In any case, if cells are running this RAD system and you put them through different inputs, resetting and setting, you can see in one axis here, green fluorescent protein intensity, they change state, they hold state, and you can keep doing this over and over and over and over again. I normally don't like to say things like this, but this is probably the best genetically encoded data storage system on the planet right now. It's 1 bit. I want 8 bits. I'll be lucky to get that by 2020 at the rate we're going. Okay, so here's the surprise pop quiz, since you're still in class. Why is it so hard? I'm just trying to control the expression of 3 different genes going up and down. How come it's so hard? How come it's such brute force trial and error? I'll make it easy, multiple choice. Few people are working to change the process. Business or political systems select for immediate answers as opposed to process improvements. It could be impossible to make biology easy to engineer, like electronics. Or there could be some other reason. I don't know what the answer is, so I'm not going to grade you. But I just want you to think about this. I'll share with you quickly my experiences, though. My experiences suggest that, for me, the answers are probably A and B. For example, when I was at MIT, we didn't have a bioengineering program, and so we were starting a bioengineering program. And any good engineer is, when you have to do something new, immediately going to look for somebody else who's already solved the problem so you can steal their solution. Right, so I found a graduate student who had earned a degree in bioengineering, and I asked for the textbook he studied. He brought me this textbook, and I started reading it to get the answers to how we should teach bioengineering. But I couldn't get past the 3rd sentence. This is going to have nothing to do with genetic engineering, apparently. There's a lot of good stuff in this textbook around medical imaging, artificial joints, and so on, but nothing too much about the engineering of biology. So that's suggesting many engineers haven't been working with biology as a partner for making things. Some of you apparently know the Honorable Joe Barton from Texas. I had the pleasure of being a witness before Mr. Barton and colleagues in the Energy and Commerce Committee back in May of 2010, along with Jay Kiesling and Craig Venter and Tony Fauci, because they were freaked out about synthetic biology, whatever that is. And this experience reminded me of answer B from the quiz. This is what we were asked. Could you engineer a genome that would predispose folks to vote for my party? It's a funny question, but it's a question that illustrates how when people turn to a technology, they typically have a problem they need solved, and they'd like the problem solved right away. Right? Now, it's also a very smart question to ask, because if the answer is yes, there's all sorts of ethics and other policy questions that need to be asked. Time and time again, when people turn to biology as a partner to solve problems, though, the problems are typically problems of last resort. They're very pressing human problems: cure a disease, save the environment, do it yesterday if you could. And if we could do it with a more mature technology that we understand better and can deploy more reliably, we do it. And what that means is the teams that then get assembled to work on biotechnology applications tend to be overdriven by the immediacy of the application and have a hard time taking even a half step back to figure out, how can I get better at doing this? And I've seen this over and over again as I've been advisors to teams working on biotechnology problems. Okay, 3rd part of the talk, tools. This is the part of the talk where I'm trying to wake up and communicate the idea that it doesn't have to be so painful to engineer biology. And that's what we've been about, in addition to doing work on data storage. It's what the BioBricks Foundation is about, many other things are about. So just to warm you up to this, I'm going to run through 2 quick historical vignettes, one with stone, the other with doped silicon. Can we make stone easy to engineer? Well, we started with things that look like this, our ancestors long ago, and we started building. Building with artifacts found in nature is quite difficult. It's beautiful when you get it to work, but you have to be an artist, an artisan. Some people become so interested in these objects that they go study them. They become geologists. But other people who like to build say, you know what? I'd like to get better at building. So I'm going to invest labor and energy to regularize the materials in this case, making standard rocks. And when that happens, you can coordinate labor across time and place and begin to assemble complicated systems, although your building blocks are ironically simpler. Like this aqueduct from Segovia, made 2,000 years ago. Others continue to get frustrated with how we're making things out of stone. So we grind up stone and we make synthetic rock, or artificial rock. You would call this concrete. But it's really synthetic stone. And when you do that and you combine that with many other tools and capacities, you can make still more improbable artifacts like this bridge in France. Or a city like Hong Kong in decades as opposed to centuries. So what are the lessons of our Stone Age? We're living in the Stone Age right now, that we might steal and use to think about a strategy for getting better at engineering biology. Well, we could invest energy to regularize the material. Could we make standard biological parts? We could get better at composing things. We could use standardization to coordinate labor, so that maybe I could work in partnership with somebody in Bangladesh or Melbourne, and they could help me, and vice versa. And I'm going to need a lot more tools. So I think these, and you could probably come up with some other lessons, are interesting to think about for the future of biotech. But what about electronics? Now this you're all more familiar with probably, right? Because in part you've lived through it. Here's a picture of a computer circa 1952. The purpose of this computer is design nuclear weapons. And it's kind of crazy. Here's a computer 25 years later. The purpose of this computer is, you would have had a hard time predicting all the things people wanted to do with computers just 25 years later. And then a generation goes by and we're familiar with today. How do you explain that over a period of 70 years you see that type of transition? It's an incredible transition. I must have some good lesson to steal from that, for sure. Well, here's an example. At the same point in time that genetic engineering was invented, these human beings are designing computers. Mid-1970s. These ladies are making the masks, apparently, for the lithography process used to manufacture patterns on silicon. You can't take these photographs today except in the context of a historical reenactment of computer design during the disco age. Right? What you would find today would be the websites for companies like Cadence Design or Synopsys, shown here, that sell billions of dollars a year in software tools that automate the design and assembly of computers. So over a human generation, we went from manual design and layout of computing systems to scaled automated design of computing systems. Genetic engineering didn't go through that transition at all. It's still largely manual design. This stack here describes fundamental process tools that have been invested in over 70 years now in support of making the process of engineering computers better. Information theory, languages and grammars for programming machines, fabrication of silicon wafers, computer design, and so on. I show you these to remind myself and you that there might be a meta lesson from electronics we could steal for biotech. And I think it's this one. When I go to buy a computer, I only care about what it will do for me. Will my phone send MMS to my mom with a photo attached? But if I want to explain how this computer exists and what the future computers will look like, I have to understand the tools layers, which are invisible and nobody really likes, except for the people who love them and make them true. But somebody's got to pay for it and sustain incremental improvements in tools affecting the design, build, test cycle so that you can get geometric improvements in what you can do at an application layer. Can we make biology easy to engineer? Don't know. Don't really want to replace the jungle, but I'd like to partner with it a lot more. It might be impossible because we've got self-mixing molecular systems that are very noisy. They copy themselves imperfectly, so they get diversity and evolution. They're very high heterogeneity. It's not like stamping out a transistor a billion times. Right? And oh, by the way, if we actually make any incremental progress on getting better at engineering biology, everybody freaks out. The ramifications of getting better with biotech are formidable. Nevertheless, I'm going to represent that over the last 10 years, people have begun to celebrate the tools at a very basic layer, and we've made progress on it. And this is where it begins to return to those dreams and the questions I want to engage on. Let me state it this way. As the tools for engineering biology improve, and people can do more in partnership with biology, how do we manage that? Can we manage that? How do we distribute that? How do we enable? How do we learn from things that might not want to see biotechnology so that we're wiser in what we do? This top row probably includes things you're familiar with, many of you. Recombinant DNA for cutting and pasting from a generation ago, polymerase chain reaction for amplifying genetic material, making a few point mutations in the sequence, DNA sequencing for reading stuff out. These are some of the basic tools that have enabled biotech thus far. Synthetic biology basically says there's a whole other row of tools to be made, and that row can be infinitely long, so long as they're useful. So synthesis of DNA, abstraction of function, and standardization to coordinate labor. You may or may not have heard of these, so I'm going to give you micro vignettes on each of them just to orient you. Here's synthesis of DNA. It starts with chemicals that were more or less worked out by the chemists around 1982. These chemicals are called phosphoramidites. They're derived from sugarcane, and they're in these bottles here. And since there's 4 bottles and we're talking about DNA, you can guess the bottles are A, T, C, and G. That's what they are. They cost $250 each. There's enough material in these bottles to synthesize 30 copies of every human genome on the planet. That's on my desk. I took that photograph. That'd be like having enough silicon to make 30x of all the microprocessors. Our machines for synthesizing DNA aren't good enough to build human genomes yet. Right? The machines just a few years ago looked like this. And what they do is they take these chemicals and a computer system will feed in information, which is the sequence of DNA you want. That controls the plumbing, so the chemicals are spit out in order. And then a de novo, from scratch, molecule of DNA is synthesized. So it's a DNA synthesizer. I view this as the most important technology on the planet. It's at least as important as silicon wafer manufacturing. It allows you to go from abstract information, to physical genetic material that, if you transform it into living systems, can direct phenotypes to the extent we know how to do that. And by important, I don't mean good or bad, I mean both. I just mean significant. That might be a better word. In 2004, the biggest thing anybody had ever assembled from scratch was 10,000 letters or bases, so a small virus genome. 6 years later, in 2010, it was 1 million bases, a microbial genome. This is what got us called before Congress with all the kerfluffle around synthesizing life. But it wasn't a designer genome, just to be careful. It was a natural genome that had been resynthesized. That's a 100-fold improvement over a 6-year period. What would another 100-fold improvement look like? Well, it'd be 100 megabases. That's quite a lot of coding capacity. That's just under the average length of a human chromosome. It's the chromosome length for any microbe I know about. It's enough coding capacity for yeast, baker's yeast.